Bueno, buenos días a todas y todos. En nombre del comité organizador local, les doy la bienvenida a la 107ª reunión de la Asociación Física Argentina. Y para nosotros es un placer ser huéspedes de esta reunión, que es el retorno a la presencialidad después de 2 reuniones que se hicieron de manera virtual. La organización de esta reunión fue bastante complicada. Primero, recién a fines de marzo, es que decidimos todos los socios hacerla de manera presencial. Y bueno, y después la situación económica del país nos hizo muy difícil tratar de estimar costos, saber qué convocatoria tendríamos, que por suerte tuvimos una convocatoria increíble. Esto no hubiese sido posible sin la ayuda de el Centro Atómico de Bariloche, el Instituto Balseiro, la Comisión Nacional de Energía Atómica, hay algunas autoridades aquí, está Mariano Cantero, Daniel Domínguez, que son director y vicedirector del Instituto Balseiro. Aquí, bueno, por supuesto, me acompaña Gustavo Monti, que es el presidente de AFA, está presente el gerente de área de medicina nuclear de la CONEA, que es Gustavo Santa Cruz. Y sin la ayuda de ellos, esto hubiese sido realmente imposible. Además, tuvimos la suerte de tener el auspicio de muchos organismos nacionales, privados y estatales, y también de organismos extranjeros. Los voy a nombrar para que vean, es una gran cantidad. Bueno, la Agencia de Promoción Nacional Científica y Tecnológica, la empresa CITES, el CLAF, el CONICET, la Fundación Argentina de Nanotecnología, la Fundación Siemens, la Fundación Balseiro, la Fundación YPF, el ICTP, Innova Río Negro, el INTI, INVAP, la empresa IOP, el proyecto Eat from Qubit, el ITBA, la empresa Mamushka, de los chocolates, la Municipalidad de Bariloche, Quantum Design, la Universidad de Comahue, la Universidad de Cuyo y la Universidad de Río Negro. O sea, a todos ellos les agradecemos un montonazo porque la verdad es que nos han apoyado para que esto se pueda llevar a cabo. Tengo solo dos anuncios muy breves respecto a la organización. Uno es que todos los participantes que no sean de CAP CONEA pueden utilizar la red Eventos, están por ahí los passwords si necesitan internet. El otro anuncio es que el edificio donde vamos a tener los pósters, que es en el edificio de telecomunicaciones, va a estar abierto por pedido de varios de los participantes de 14 a 19, o sea que a partir de las 14 ya se puede ir a colgar los pósters. Y bueno, creo que eso es todo. Cualquier inconveniente que surja, por favor, no duden en contactarnos. Somos los que tenemos la credencial de este color, todo el comité organizador. Por supuesto, me olvidé decirlo antes, esto fue el trabajo de muchísima gente, todos los miembros del comité organizador local, los miembros del comité científico, que fue coordinado por la doctora Gladys Nieva, así que a todos ellos un agradecimiento enorme. Así que, bueno, creo que nada más de mi parte, espero que tengamos todos una excelente reunión y los dejo con las autoridades del Instituto Balseiro.
Buenos días a todos y todas. Bienvenidos, bienvenidas al Instituto Balseiro en nombre del Instituto, la Comisión Nacional de Energía Atómica y la Universidad Nacional de Cuyo. La verdad que es una gran alegría para nosotros recibir esta reunión acá, la 107ª de la Asociación de Física Argentina. Un agradecimiento a Diego, a todo el equipo organizador y en nombre de él a todos los que colaboraron. La ciencia y la tecnología son pilares fundamentales para el desarrollo del país. Todos los países que se desarrollan y que crecen y que terminan teniendo una mejor calidad de vida de sus habitantes lo hacen en aportes al valor agregado que es la ciencia y la tecnología. Ahora, lo que tenemos que tener claro es que la ciencia y la tecnología son sus investigadores, sus investigadoras, sus docentes, sus estudiantes, no son las grandes instalaciones, son los recursos humanos que hacen que eso produzca y que eso impacte. Así que eso lo tenemos que tener muy claro. Reuniones como estas, en las cuales la gente se junta, aporta, se le da también rumbo con vistas a mediano plazo y largo plazo, son cosas fundamentales que tenemos que proteger y trabajar juntos para poder cuidar nuestros recursos humanos, que es lo central y que lo que nos va a asegurar que esto pueda seguir funcionando. Es una alegría muy grande. Estaba hablando con Gustavo, que fue profe mío en el 94, en el FAMAF. Así que seguramente ya he encontrado otros amigos, a Jorge también, coterráneo de Río Cuarto. Es una gran alegría verlos acá con toda esta actividad. Van a inaugurar el edificio de Teleco casi, digamos, no inaugurar porque no está todavía, le falta algunas cosas, pero va a ser la primera actividad formal que ocurre en lo que tiene que ver con ciencia y tecnología, educación en el edificio. Un predio muy lindo que hemos logrado terminar. Y finalmente quería hacer un pedido. Es que muchos 7 de junio circulan saludos por el Día del Físico, la Física, y formalmente entiendo que eso no está oficializado. Así que el pedido era que la asociación en la asamblea que tenga discuta eso. Posiblemente pueden terminar haciendo un pedido de que esa fecha se oficialice y después vaya un pedido de la AFA a que el Congreso lo sancione como ley, el Día del Físico y la Física en Argentina. Así que bueno, con eso cierro. Una gran alegría ver toda esta actividad, ver toda esta gente. Fueron 3 años durísimos de pandemia y ver el salón de actos lleno y la plaza esta que recientemente pasó a llamarse Plaza José Antonio Balseiro, donde está la dirección del instituto, los restos de José Antonio Balseiro, un retoño del manzano de Newton y 3 arbolitos en memoria de 3 desaparecidos que tiene el instituto en dictadura. Verla llena de los estudiantes que cruzaban al aula, la verdad que nos llena de alegría, de esperanza y de satisfacción. Así que bienvenidos, bienvenidas. Espero que sea muy productiva esta reunión.
Hola, bueno, buen día. Bueno, también bienvenidas, bienvenidos. La verdad que es una alegría tenerlos acá en nuestra casa. Y bueno, y volver a tener una reunión de la AFA presencial, volver a abrazarnos cuando hacemos la cola para la inscripción, a saludarse, a reencontrarse, realmente es maravilloso. Así que contento de un montón de gente que no veía de hace años. Estamos igual o hasta más jóvenes. Y también, bueno, muchos estudiantes nuevos que también han venido. Así que, bueno, bienvenidos. Espero que la reunión sea tan buena como parece leyendo el libro de resúmenes y las charlas plenarias. Así que, bueno, adelante. Y bueno, le paso la palabra a Gustavo. Bienvenido otra vez.
Buenos días a todos, amigos, amigas. La verdad que es una inmensa alegría que hayamos podido volver a estas reuniones. Debo ser honesto, yo tenía un miedo, digo, la primera presencial se hace en Bariloche, queda lejísimo, los fondos no alcanzan, ¿cuánta gente habrá? Y la sorpresa fue inmensa. Cuando Cecilia Ventura me pasaba los datos, ya tenemos 300 inscriptos, ya tenemos 500, ya estamos pasando los 600. Digo, bueno, zafamos, todo un éxito, como no podía ser de otra forma. La gente quiere mucho, Bariloche quiere mucho al instituto. Así que les voy a pedir que asistan a la asamblea, porque ahí les vamos a contar lo que pudimos y no pudimos hacer. Los 2 años de pandemia, la verdad que fueron una traba importante para la asociación. Las reuniones de la asociación, si bien las logramos hacer virtuales, bueno, claramente, por todo lo que se ha dicho, no es lo mismo. No satisface todas las expectativas de los socios y socias, así que hacerla presencial es como volverle a dar más del 50% de vida a las actividades de la asociación. Así espero que todo sea un éxito, estoy seguro que va a ser un éxito. Tenemos elección de autoridades, aparte de todo. Así después de 6 años que tuvimos en la cabeza la responsabilidad de dirigir la asociación, bueno, ahora toca el cambio de autoridades. con una muy buena perspectiva de género en la nueva comisión directiva. Y bueno, simplemente, de vuelta, ya agradecieron Diego, ya agradeció el señor director del instituto, los mismos agradecimientos para todos. Inmenso agradecimiento al instituto, a la Comisión Nacional de Energía Atómica, a la Fundación Balseiro, a todos los que han colaborado, todos los que han participado del comité organizador. Sé que han tenido un trabajo muy, muy duro y, como decían, como decía Diego, con mucha incertidumbre, ¿no? Saber, conseguimos los fondos, no conseguimos los fondos, la situación está difícil. Tenemos que volver a la presencialidad. 2 años de discontinuidad es como que todo se desorganiza y tenemos que empezar casi de nuevo. Así que inmenso agradecimiento a las divisiones de AFA. Las divisiones han estado muy activas en la virtualidad también. Han hecho hermosas cosas y creo que les da mucha vida a la asociación la actividad de las divisiones. Las divisiones son como pequeñas asociaciones en sí mismas que generan muchas iniciativas, mucho trabajo y aglutinan mucho a la gente. Así que es muy, muy importante para nosotros la actividad de todas las divisiones de AFA. La subcomisión de género, un inmenso trabajo. Muchísimas gracias. Todos los años su informe, un informe muy prolijo, muy amplio, una gran actividad la subcomisión de género. Así que también a ellos un gran agradecimiento. Y bueno, no voy a seguir contando más, el resto va a estar en la asamblea, todo lo que hemos hecho durante estos años, así que los esperamos por ahí. Muchas gracias a todos y a todas.
Empezamos entonces con la presentación de la primera charla plenaria. ¿Cómo?
So, good morning, everybody. And it is an enormous pleasure for me to be here today introducing Professor Jean-Louis Barat as our first plenary speakers of our National Physics meeting, finally presidential after 3 years. So I'm here representing Soft Matter Division and its members who are grateful to Jean-Louis for kindly accepting our invitation and his presence here today. Thanks to all his collaborators and international fundings shared with researchers from here, from Centro Atómico Bariloche, and from our south in Universidad Nacional del Sur in Bahía Blanca. So I would like to tell you a little about Professor Jean-Louis. He was born in Paris, in France, and he had his studies in Paris 6 and the École Normale Supérieure, where Pierre Deschamps, a well-known reference in soft matter physics, was the director, obtaining a degree in physics in 1985. Just 2 years later, he obtained a degree, the doctor in theoretical physics at Paris 6, and afterward he held 2 years postdoctoral position at the Technical University of Munich in Germany, and a second one at the Department of Chemical Engineer in Santa Barbara, USA. From 1994 until 2010, he was full professor at the Université de Lyon, where he was later director of the Laboratory for Condensed Matter and Nanoscience for 3 years. In the period 2014-2021, he was director of the Laboratory for Interdisciplinary Physics at Dual Dependency Institute from Grenoble and CNRS. In turn, since 2011, he's full professor at the University of Grenoble. And since 2012, he's an active member of the theory group of the prestigious Institut Langevin in the same city. He's selected as director of his group in 2020. And on research, Professor Barat has contributed to multiple lines in physics like liquid, crystals, polymers, granular matter, interfaces, and nanostructured materials. And he has contributed to the solution of theoretical problems in statistical physics and the development of new computational methods for many physical problems. Professor Barat is internationally recognized for his career and scientific production, having co-authored almost 200 papers and 2 scientific books, which give him an age index of 75. In addition, Professor Barat has received 15 important awards and distinctions throughout his career and has served as an editor in important journals of international relevance such as PRL, EPL, Chemical Physics, and Journal Physics Materials. Even more, Jean-Louis, with her well-known kindness, has received as visitor many students and researchers from Argentina in his different group, and he has helped to the formation of our young researchers and students and his future. So, having said all that, it is a great pleasure to leave you with Professor Barat, who is going to give a talk on flows of materials or the formation of amorphous solids. And so thank you very much, Jean-Louis, for being here today with us in Argentina.
Thank you very much for this introduction. It's a great pleasure to be here again in Bariloche, and after a few years without traveling, as you know. So today I would like to give you some perspective on work that was done mostly in Grenoble over the last 10 years about deformation of amorphous solids. So as often, this is the work of a rather large international team, and as any good French team, it has a few Argentinian players, so you might recognize them here. And I would especially like to acknowledge the very important contributions of Kirsten Martens, who is now a CNRS researcher in Grenoble, Alexandre Nicolas, a former PhD student who actually was also a postdoc here in Bariloche and is now a researcher in Lyon, and Ezequiel Ferrero, who is an investigator here in CONICET in Bariloche. So let me first define what I will be talking about, what are amorphous solids, especially soft amorphous solids, and why is it interesting to investigate their flow properties. Well, amorphous materials, amorphous solids, come in many different species, and they actually go from very hard systems like metallic glasses, window glass, or polymer glasses, to very soft systems, those that you might not usually think of as solids, but that actually are solids, like foams and gels, like here, toothpaste, probably. And so all these systems are solids in the sense that they do not flow unless you apply a strong stress to them. In terms of their elementary constituent size, they span many orders of magnitudes, from molecules here to bubbles here that might be, so here this is sub-nanometer, here this is millimeters. But they share the important properties that, as I said, they are solids, elastic. And here I will only consider systems that are below any temperature at which they would start to flow spontaneously. So I'm not sure what I did with this. I will just remove it. Okay, so as you know, the glass transition is also so the transition from a liquid to a disordered solid. This is also a very important topic in statistical physics, and the last year Nobel Prize was awarded to Giorgio Parisi in part for his work on this topic. But this is not what I will be talking about today. What I want to talk about today is really taking such a solid system and applying a stress to it so that it deforms and eventually flows. Okay, so, sorry. So the flow of these systems can be characterized in two different ways. You can apply, so I will be thinking of the basic type of deformation, which is shear flow here. What I'm saying applies to other types of deformations, but to keep things simple, let's have in mind that you take a system like a foam, or maybe like a metallic glass, but you shear it in simple shear as here. And so then your control parameter is the deformation rate, so the velocity at which you shear, essentially. And what you will plot in a steady state is the stress, so the force that you have to apply in order to obtain a certain velocity, a certain shear rate. So the typical flow curve is represented here. So this is the steady state stress as a function of the velocity in a log-log scale. And you see that because your systems are solids, you need a minimal stress, which is called the yield stress, in order to create flow. And if you are above this minimal stress, then you typically have a stress that increases with the flow velocity. And this increase is often characterized empirically by this Herschel-Bulkley equation. So the stress is the yield stress plus some power law of the velocity of the strain rate. Another possibility is to characterize your system with what is called a stress-strain curve, which is essentially you start from a system at rest, and you start applying a deformation to it at some fixed rate. And so first, you will observe elastic behavior, essentially up to this yield stress here, and then you will observe the flow. So 1 point on this plateau here for a given strain rate corresponds to the value of the plateau here corresponds to 1 point on this curve here. When you look at things on a more microscopic scale, which you can do using computer simulations, but which you can also do in experiments, for example, by considering a foam in which the number of bubbles is relatively small, so you essentially have access to something that has only a few atoms. If you identify a bubble with an atom, then you see that this stress-strain curve that I was showing here is actually not a smooth curve. It consists of an elastic part, so a loading here, the stress is proportional to the strain, so this is the Hookean solid. And then in the part that at the macroscopic scale looks like a plateau, a constant plateau, if you look with a very high resolution, you will find that this actually consists of parts of elastic loading separated by strong drops that come in all kinds of scales here. Okay. And so this behavior is the one that will be of interest in the following part of the talk. So where does this come from? This was understood first, probably in foams, that when you deform a system, the deformation does not proceed in a smooth way at the microscopic scale, but it proceeds at the microscopic scale by very well identified events, which are called plastic events, that in a foam will correspond to a change in the local topology. So here, if you follow these 4 bubbles in the foam that is being deformed here, then you will find that during the deformation, bubbles B and D, that were neighbors at the beginning of the event, eventually exchange their neighborhoods and their topology, and then at the end of the events, A and C are neighbors, and D and B do not share an edge anymore. And this happens only in a rather localized part. So if you would look in detail at the topology of the system around this special event here, it remains unchanged. So of course, there is a small deformation if you follow the bubbles here, but the topological change is really localized here. And this is how the deformation proceeds in foams. And it has been shown later on in many experiments that you have the same kind of pattern in all kinds of disordered systems, here a colloidal paste, essentially. The events are more complicated and not as easy to describe as the topological event in the foam, but basically they have the same nature in the sense that there is some topological change in a very localized place, and around it, the system just deforms smoothly. So this is best, so these events, more generally than T1 events for the foam, are generally called shear transformations, and they can be described as a local instability of a region, of a well-localized region of the system, in which under the influence of the external deformation, the system, the particles that were localized in some energy minimum undergo a transition to a different energy minimum locally close. This typically involves a few tens of particles, and it is a very localized event in the sense that the topological change of this irreversible behavior that is illustrated here only occurs for these few tens of particles, and the surroundings do not undergo this kind of irreversible behavior. They just respond as a homogeneous elastic medium. So this kind of response to an elastic medium, to a local irreversible transformation was described long ago by Eshelby, who is well-known in metallurgy. And you can think of it as a circular region of your system here, undergoing a transformation in which it suddenly, because it becomes unstable due to the external deformation, it suddenly wants to become, let's say, an ellipsoid. And then it will exert, if this happens, then this inclusion here will exert forces on the surroundings, and these forces are essentially two dipoles of force perpendicular to each other. So then what Eshelby calculated in detail is the response of the surroundings to this kind of change of the local, let's say, ground state of the inclusion. And the change can be described in a 2D system by this propagator. So the system around the inclusion will actually deform according to a quadrupolar symmetry, because you have these two dipoles, and the dipolar power law here, 1 over r squared. So the fact that these are the elementary events that govern the deformation, this has been studied by many groups in experiments, in simulations, and it's quite well established. The kind of experiments that allow you to best characterize this kind of behavior are really correlation experiments in which, for example, here you do a deformation, a cyclic deformation of a colloidal glass. So here, these are the state for deformation, and you come back here. And then you look at the displacement of the particles in this deformation. So these are the displacements measured by confocal microscopy. And in the displacement, you do not see much, in fact. But then if you do a correlation of images that are separated by one period, so you do sort of a stroboscopic experiment, then you find very clear correlations in the displacement that can be shown to essentially reproduce this Eshelby pattern. So it's quite well established that the events that will take place when the material is deforming, they are correlated, and they are correlated due to this elastic interaction. Now, when you have correlated events, when you have events that interact by such a long-range elastic interactions, what will happen at large deformation is that these individual events, so these shear transformations, will actually interact, and they will organize in the form of what is called avalanches, in which one event will trigger events in the other parts of the material, and eventually you might end up with something I would call a big, very big earthquake, so just a big event that spans the whole material. Okay, so this behavior that is very well known in many condensed matter systems with disorder, in the magnetic domain walls that are studied very much here in Bariloche, contact line, vortices, and it can be, I mean, this kind of picture can be scaled up to describe earthquakes, friction, etc. And the usual framework in theory to describe it is that of an elastic line pinned by some external potential. Here, so let me conclude this brief introduction by introducing the flow of amorphous solids, which is also, I should mention it, it's also a domain that has many applications in rheology, in material science. But here I would really like to consider it as a statistical physics problem in which we have well-identified elementary events, these shear transformations. These events will interact in space and time and become correlated. One interesting observation is that the flow curve that empirically is written by people doing rheology at the stress is the yield stress plus some power law of the strain rate. This can be rewritten by people doing statistical physics at the strain rate being proportional to the stress minus the yield stress to some exponent. And of course, this looks like a dynamical phase transition behavior. The features I was describing are now believed and it's quite accepted that they are very universal. They will govern the deformation of all kinds of disordered media, from granular media to metallic glasses. This situation is probably quite different, and perhaps one could argue it's a bit simpler, that the situation from crystalline plasticity, so the deformation of crystalline solids, that involves well-identified flow defects such as dislocations. So what I would like to do now is to argue that one can try to gain qualitative understanding of the phenomena with relatively simple models. And then one might tune the models to make quantitative predictions on specific systems. And there are several scales at which one can attack the problem, a microscopic one, a continuum one. I would like here today to concentrate on a sort of mesoscopic scale in which I will use the shear transformations, which already involves maybe 10, maybe 100 atoms locally, as elementary events. So I coarse-grain, I forget in a sense about the atomic scale, and it will take these shear transformations as elementary events with elastic interactions between them. So this is the center of what we call elastoplastic models of flow, in which I I should switch my Wi-Fi on because it's doing strange things. Okay. So elastoplastic models, I would like to describe in the sense as analogous to Ising models for deformation of disordered system. In the sense that in Ising model, you forget about the origin of the exchange interaction, you just concentrate on the spin. Here, in the same way, we will build models in which we divide the system into, let's say, sites. Each site has maybe 100 of 1,000 atoms, here bubbles, in fact. And on each site, you have the possibility that the system undergoes a shear transformation, okay, under the external strain applied. Then, if the system undergoes a deformation locally, then this part, in a sense, becomes unstable. So it cannot sustain any stress, it cannot sustain the stress anymore. And so the stress that was supported by this part has to be redistributed on the neighboring sites, and it does so with the elastic interaction I described before. So now, once you have understood these elementary ingredients, you can build a very simple model. And here, to make things simple, we keep the stress as a scalar variable. Of course, you can make it a tensor, etc., but it doesn't really, it makes just things a bit more complicated, but here I will stay at the scalar level. So on each site here of my system, which again, this site can be a big region of the material, or I mean, mesoscale region of the material. On each site, I have a stress variable. So this stress variable will increase with time. As I apply an external shear rate, it will increase proportionally to the external shear rate. So this would just be the Hookean elastic solid. Now, because you have this possibility of local failure here, local plastic activity, then the local stress on site I, let's say, will also increase because of the contributions it is receiving from sites that are actively plastic here. So if J is the site here, site I will receive some extra loading because J is actually failing at this moment. And the propagator that carries this information will be this elastic propagator here. So now this one has to, this part will be quite universal. And so here I'm just saying, basically, if J is undergoing a plastic event, so it's undergoing this type of shear transformation, then the local plastic strain in J will be non-zero, and it will be proportional to sigma J, but this doesn't matter so much. And if the site J is elastic, then this part is zero. Okay, so that's, and now when does the site J becomes plastic, sorry, when does it undergo this plastic activity? Well, it's when the local stress sigma J becomes larger, sorry, there is a, so this should be larger, sigma J is larger than sigma Y, which is some local variable with some disorder. Okay, so then the site will become elastic again after, let's say, a typical plastic deformation on site J. So after you have undergone the, let's say, the topological change locally, you are in the new minimum and you become elastic again. And so this kind of model can be very easily implemented on a lattice, and it can be studied computationally at a relatively moderate computational cost. In contrast to, if I wanted to do a full atomic simulation of the deformation, this is extremely costly. Computationally. Okay. So let me just very briefly state that we are able to validate the approach against experiments that are done, for example, on microemulsions, so basically foams. So using this kind of model, we can reproduce the flow curve, we can reproduce different flow patterns in different geometries, and we can also reproduce fluctuations in the flow. So we are quite confident that these kind of models, if you calibrate parameters properly, can be quite well, can be used to describe the flow of, for example, a foam or a paste. So now, let me go back to the statistical physics perspective. When you have such a simple model, the first thing you want to do is a mean-field analysis. So mean-field analysis means that, as I was mentioning you, on every site here, I have defined a stress variable, sigma I, sigma J, etc. And so what I will try to analyze is how the probability distribution of the stress on a given site evolves with time. Okay? And the mean field means that I will be considering a typical site, okay, in a flowing medium, okay, and consider the probability distribution of the stress of the stress variable on this given site. So the midfield analysis goes as follows. If I take the time derivative of this P of sigma t, so the probability distribution of a stress on the given, on the specific, on the typical site, then it will first increase due to the external loading. So this is a usual sort of a convection term in the space of stress. And so it's proportional to the external loading gamma dot. This is the elastic part. Then let me identify the other terms in these equations. Well, first of all, I will have one term here, the third one, that tells me that the stress, that the site will yield if the stress becomes larger than some critical value. And so this will be a loss term with a rate that I will describe later. Then, when the stress yields, I will assume that its stress, when the site yields, I will assume that the stress is reset to 0. So this gives me a gain term at sigma equal to 0. And I will come back to the prefactor. And then there is an additional contribution, which I will describe as stress diffusion. So a given site is being loaded externally here, but it's also receiving permanently kicks from other sites that are plastically active. And I will describe, so these kicks can be positive or negative, because as you have seen, the elastic interaction is anisotropic, and it's cosine 4 theta, so it can be positive or negative. So if you receive positive and negative kicks, the first reasonable assumption is that you will undergo a diffusion process. And this is described by this term here with the second derivative with respect to stress. Now, in order to close the equation, you have to specify what is the yield, what is the rate here at which you yield. So basically, you yield if you are above sigma c here, and you do so with a rate which I will call 1 divided by tau here. And then the yield, the rate of gain at 0 is, of course, all the sites that are yielding are being reset to 0. So this is given, so this rate here is given by this integral over p here, okay? And finally, the assumption will be that the kicks that you are receiving here, so the diffusion coefficient in the stress space, they will be proportional to the number of sites that yield per unit time, which is just this capital gamma of t that is here. So you see this Midfield equation, it's not very complicated, but it is a nonlinear equation in P, because so these two terms here, these three terms and 4 here are linear in P, but because the stress diffusion here depends on the plastic activity, then this becomes a nonlinear equation in P. Still, even if it's nonlinear, it can be solved in a steady state exactly, and the result it yields is that if the coupling here, alpha, is large, then you get essentially a liquid. If the coupling becomes small here, you get a Herschel-Berkeley law with an exponent 1/2, which is typical of Midfield approaches, if you could argue. And so you get this critical behavior that the stress increasing as the yield stress plus the strain rate to the 1/2, the square root. So this equation has been studied a lot, including by mathematicians, but I will not detail it much more. I mean, it's satisfactory that with such a very simple approach, you get something that is not so far from experiments. What I forgot to mention is that in experiments, the exponent that is found here is well, typically around 0.6, 0.7. Okay, it's not determined as accurately as in phase transitions, but the order of magnitude that is reported by experimentalists is of the order of 0.6 to maybe 0.8. So it's satisfactory to have a very simple midfield approach that reproduces this basic phenomenology. Now, if one wants to go beyond midfield, and this is something you would like to do, because first of all, you see that the exponent is not the one that midfield is giving, but it's something different. And also, you have this critical behavior, so you know midfield is not usually satisfactory for describing dynamical critical phenomena in 2D or 3D. Then you can attempt a scaling description, and this was done by Mathieu Viard, Alberto Rosso, and co-workers. And in fact, the idea behind the scaling description is very similar to the one that is used in deepening theory, so the theory that is used to describe, let's say, the motion of a magnetic wall. So in deepening, you have something which is very similar, that if you apply, let's say, a magnetic field to a magnetic domain, then the velocity of the wall of the magnetic domain will be proportional to the applied force. And it will be will increase with the applied force, but only if the applied force is above some threshold value, and the velocity increases like f minus fc to some exponent. But now, what is found in deepening problems is that beta, the exponent here, is smaller than 1. In the yielding problem that I was describing, we have a similar behavior formally, that the strain rate is proportional to sigma minus sigma c above some yield stress sigma c. But now, systematically, the exponent beta is larger than 1. So we already know from that yielding and deepening are different phenomena. What is the major difference between them? Essentially, the major difference is that in deepening problems, so when you have an elastic line that you pull on with a force, and you try to move it in a disordered energy landscape, let's say, then when the line advances somewhere, it will always increase the force on other sides. So the elastic interaction in the line is always positive. Now, with the elastic kernel I was describing, then G of R minus R prime, this Gij, is not positive everywhere. So you have, instead of having a systematic force that is pushing you always in the same direction, you have this diffusion term that I was describing in midfield, and this gives rise to a new type of universality class, in a sense. So so this still can be studied in detail. So as I was mentioning, when you are in the steady flow regime, you have this interaction between events, and these earthquake or avalanche-like events have a behavior that is very typical of critical phenomena. So they are distributed according to a power law. So the size of the drops, or the size of the earthquakes, if you like, is distributed according to a power law. And this power law is cut off only by the system size here. So that's very typical of critical phenomena in general. And this is the behavior that you observe when you pull a system slightly above the threshold value of the stress. And so this can be observed in all kinds of simulations, but the best determination of critical exponent is by using these elastoplastic models, because they are those that allow you to go to large sizes. So I will not ask you to or comment all the work that can be done on critical exponent, but I would just like to mention that there is no complete theory of exponents for this kind of critical phenomena. There are quite well-established scaling relations. There is a very well-established notion that at the beginning of the flow, what you have is what is called the marginally stable solids, in which you have many points that are on the verge of yielding. And these can be used to establish scaling relations, but there is no detailed theory of critical exponent. So now, let me say a few words about a more slightly different situation. In fact, what I described until now is a situation in which the stress always increases with strain rate. And this gives rise to this avalanche behavior, second order type of dynamical phase transition. But it turns out that experimentally, it is observed that in many cases, what you observe is a rather different picture. It's not second order in a sense. It's a situation in which you can have in the same system coexistence of regions that are flowing and regions that are staying in the solid state. So these are several different examples from different systems, bubble raft, chocolates, granular paste, etc. And this is an example from simulation. So what is shown here is basically in a sheared system, the value of the velocity as a function of position. And so here you see you have one part in which the velocity is non-zero, so the system is flowing, and this part is coexisting with a part in which the velocity is zero. So and this is for the same value of the stress. So this is just a first order type of situation in which you have regions with different properties, like solid and liquid, that are coexisting in space. This situation is called shear bonding or strain localization, and it can be understood from the fact that the systems that have this kind of strain localization of shear bonding have a flow curve which is not of the Herschel-Bulkley type as I was describing before, so not a monotonous flow curve, but they have actually a flow curve that is shown here in brown. Okay, so it is first, you have to increase the stress up to a yield stress in order to initiate flow. But then the flow curve is not monotonous. And it means that for the same value of the stress, so just applying the same force, you can have two different velocities coexisting, zero velocity here, and a finite velocity here. Of course, there is another point here, but it's easily shown to be unstable here, okay, the intersection here. So you have just these two phases, in a sense, solid and liquid, that can coexist over a range of stresses here, forces. So this, again, can be understood using the kind of models I was describing. For example, if you take as a rule here that one element yields, but remains, when the element yields, it remains damaged for a long time. So this tau res is the time it takes before you recover elastic behavior in your element, in your local element. It turns out that if this time is large, then you can understand that the deformation actually will sort of concentrate in places that have started to yield in the first place. And this is essentially what happens. So using these elastoplastic models, one can show that if the time here for recovery or healing locally is large, you get these non-monotonous flow curves, and you get strain localization. Okay. So here this is a system that an elastoplastic system like the one I described before. You start to shear it from a system at rest, and then you find that the deformation will localize in one specific part of the material. And then it will stay there, and you will have solid material coexisting with liquid, in a sense, material here. Good, let's skip that. Let me conclude by saying that there are other possibilities for shear bonding, and I would like to conclude on this part by saying that the flow of soft solids can be described by these simple models. It can be the transition from solid to flow can be in some cases a second order dynamical phase transition with a different universality class than depending, also it's somewhat similar. But there is also the possibility of a first order like transition. And I would like to finish here by extending this type of ideas to systems that apparently are very different from forms, but in fact we will see not so much, which are living tissues. This is mostly work that was done in Grenoble by Daniel Matos Fernandez, who is now at the University of Warsaw. And the motivation for that is in the sense that people are now able to study the properties of tissues using the same kind of tools that they use for studying the properties of colloidal systems of foams with imaging, tracking, etc. And so you can follow here the life of a tissue in which cells are dividing and dying in time. So what we would like to do is to try to understand in such a system that is a living system in which you have death of cells and division of cells and perhaps propulsion of cells, can we make some analogies with the kind of soft materials I was describing before? So here I will go back to a particle-based model. And so basically this particle-based model is very simple. It's just soft spheres with overdamped dynamics. And the only new ingredient that I will put is that these soft spheres have the possibility to die. So they will undergo what is called apoptosis with a rate that is well defined. So the cell die with some given rate. They also have the possibility to divide, at least if they do not have too many neighbors. So there is also a division rate. So the system is very simple. It's a toy model in a sense. You have very few parameters. You have the attraction between the spheres. You have the division rate and the rate of the apoptosis to the division rate. And so with this, if this rate here is small enough, so if you do not die too fast, then you reach a stationary state, which is called a confluent tissue, in which the system is stationary statistically with death and division of cells at the same rate. So now you see that the division of, or the death of a cell, in a sense, it's expected to play a role which is quite similar to the T1 events in foams, except it does not depend on an external driving. Now, it's an internal motion of the system. And so this is seen, for example, by looking at the strain-strain time correlations in the tissue in the same spirit as the experiments I was showing in the beginning. So if you look at this strain-strain correlation, you see this kind of pattern appearing, which is characteristic of elastic response to a force dipole, in a sense. So now the question that we asked was, what is the consequence for rheology? And basically, I should say the system, because of these events, always remains a fluid, in a sense. So it's never a perfect solid. It's never an elastic solid. It always flows at very small strain rates. And so you expect some kind of crossover between a liquid-like behavior at small strain rate and that high strain rate, the same behavior I was describing for soft solids. So this is, in fact, what we see in doing simulations of the model. So these are the flow curves in which you have a liquid-like behavior at small strain rate, and then you cross over to the soft glass or soft solid behavior. And I will go fast, but I would just like to say that all the rheology of the system can be essentially understood by generalizing the analysis I made for the soft glass by introducing an additional diffusion here that is related to the birth and death of cells. So an additional stress diffusion. Yeah. And this allows us to reproduce quantitatively all the curves that we obtained from simulation. So I think my time is over, so I will just stop here by saying that this is a very simple model of a living tissue in which we try to extend the ideas that are coming from the soft glass studies that we did before. And it can be described by a simple midfield model quite quantitatively, I mean, at least not in comparison to a real tissue, but in comparison to the simulations that we can do. And I would like to thank you for your attention. Sorry, a little bit.
Jean-Louis, as always, perfect on time, an excellent talk. So the session is open for questions. Somebody?
Thank you very much. I have some echo. Impressed about the implications of those approaches to living tissue. But my question is, there is some important issues related with very metastatic and malignant tumors that are embedded in normal tissues, that also tissues that are subject to mechanical stresses. And somehow the normal tissue can force the tumor to spread tumor cells in the normal tissue, and the metastatic effect will be greater. And so there are some theories I asking you about modeling that behavior, but in the interface between tumor and normal tissues.
I'm not an expert in this field, but yes, there are many people trying to understand the interplay between cellular division and mechanical stresses. And it's actually becoming a whole field. I know a number of people in France doing that, like François Gardner, Jean-François Joanny, and also doing experiments, actually, so trying to induce mechanical pressure on tumor cells and yes, actually in my institute in Grenoble, there are a few teams. There is one team working on that. We had some interactions, not to the level of completely modeling the experiments, but yes, there is a lot of interest in it.
So thank you. Somebody else? Please.
Sorry?
Thank you very much. There have been many advances in the last years in understanding thermodynamics on the microscopic scale through fluctuation dissipation theorems and things like that. Can you observe these things in your experiments and your simulations?
I mean, we did study these type of issues, fluctuation, dissipation, etc., a lot in the rather high temperature regime. So when you take a system that is basically close to the glass transition, close to the region between glass and liquid, and then you shear it, etc. In this kind of system, the systems are really thermal in a sense. And you have something that you can describe as mechanical noise. But this is not, because you have, you get these kicks, so you could say this is a bit like thermal noise. But it turns out the properties of this noise are very different from the properties of thermal noise. So in that sense, there have not been that many studies on fluctuation, dissipation in this system. I mean, because you cannot really define well the effective temperature or something.
Yes, true. It's okay for you to answer. Somebody else? Any other question? If not, I have one, Jean-Louis. You mentioned the diffusion at the last fields on tissues. It was the only parameter to free parameter to compare with experiments. Are you trying? Did you try something with experiments or some comparisons? Or I would like to know at least the order of magnitude of that.
No, I think the model is really probably too simple.
All right.
Okay, but try to force it perhaps. But it's also very difficult. I mean, the problem is that the time scale for apoptosis is rather long.
Yeah, I know.
And so comparing, doing, for example, rheology experiments on this scale,
that's very hard.
That would be really hard, I think. Especially with real biological systems that have all, they are living systems.
Okay, great. Thank you very much. Any other?
I have a question. So most of what you explain depends on this event where this sphere transforms into some kind of ellipsoid. So my question is this just the only kind of event? So aren't there any other like topological formations like Koster-Taules or something else that enters into play to describe the different phases of these disorder systems?
We believe not. Well, I mean, I cannot, of course, be. First of all, it's very difficult to identify the order or topological defect. So you just identify the events, but associating the event with some local geometrical property is very difficult. I mean, it just takes place where it takes place, and you may find criteria to. And then my answer would be, if with these simple models we find that we can reproduce experiments and we can reproduce also simulation, detailed atomistic simulations, etc., then probably there is no need to introduce something more complicated. But of course, I cannot exclude that in some systems you have more complicated types of events taking place.
So thank you. We have time just for a short question. We are just on time, almost 11. Somebody else? If not, I think that it's time to thanks again, Jean-Louis, and its excellent talk.
Gracias. Buenos días. Siguiendo con las sesiones plenarias de esta mañana, tengo el placer de presentar a la doctora Alexandra Rakovich. Voy a continuar con la presentación en inglés. Dr. Alexandra Rakovich is a senior lecturer in the Department of Physics in King's College London. She's also editor of the Plasmonic theme in Frontiers in Plasmonic Journal. She has finished her PhD in 2011 under the supervision of Professor John Donegan in Trinity College in Dublin, Ireland. Her PhD work was focused in the interactions of colloidal semiconductor quantum dots with various biomaterials, such as membrane proteins and photosynthetic reaction centers. Then she moved to the physics department in Imperial College in London, where she worked as a research associate for 4 years with Professor Stefan Meyer. Before transitioning to the Royal Society University Research Fellowship, in 2017, she joined the physics department in King's College London, where she is a research fellow and lecturer, and she's currently working there. Her main research interests are biomimetic nanophotonic systems, the use of nanoparticles for biomedical applications, nano-enhanced biophotonic materials, light-matter interactions in plasmonic systems, and the control and localization of metamaterials. We have a very fruitful collaboration with Sasha, which is, we have a joint project with the Royal Society of Chemistry and CONICET, and we are collaborating in the development of new plasmonic substrate with hybrid antennas for the development of SPR and source-based mixed technique, which I will present in the Optics Center. So I'll let you with Sasha for her conference. It's a pleasure to have you here.
Thank you, Laura. I'm not sure if the mic is working, but everybody can hear me? Okay, great. Laura, thank you very much for the introduction. It's an absolute pleasure to be here and to visit Bariloche again after 4 years of isolation and being in UK, essentially not being able to travel, to see people and to present in front of an actual live audience instead of a blank screen as we usually do. So as Laura mentioned, I am from King's College in London. So it is a college that consists of many different departments and many different campuses, in fact. But we are located in central London, just here, which is about a half-an-hour walk from Buckingham Palace if you wish to visit for afternoon tea or something. King's College is also part of something called London Center of Nanotechnology, which is a center where you can do a lot of nanofabrication. And it's a joint venture between several colleges in London, but we do make very strong use of it, and some of the research I presented was essentially done in that center, which is why I wanted to acknowledge it straight away. So within King's, the physics department has several directions, mostly along the biophysics, soft matter, and also particle physics, cosmology, astrophysics directions. But we also do have this group of photonics and nanotechnology, which is where I am based. So a lot of my work is to do with photonics, but I also take opportunities to collaborate with other groups within King's and elsewhere to work on nanophotonics for bioapplications. So this is more nanomedicine type applications. We also have a strong direction working in the nanophotonics for clean energy sustainability. So that's more along the light harvesting directions or photocatalytic applications. And finally, a lot of the work in both directions is actually underpinned by my third direction, which is what I will be talking about today. And this is nanomaterials assembly and their control. So why do I like to work with nanomaterials, and why specifically I would like... No problem. Oh. Okay, better? Okay. Yes, I do speak quietly, apologies for that. Okay, awesome. It's good. Yes, okay. All right, so I do like to work with colloidal nanomaterials because I believe that they're the direction in which we should go if we were to consider full technological implementation of nanomaterials and also being able to extract the most use out of them. And the reason for that is that, for example, there's two types of producing nanomaterials, and one is a bottom-up method which produces colloidal nanoparticles in solutions. And the other one is top-down approach, which is essentially lithographic methods where you deposit films on substrates which are nanostructured. And the problems, both of these have their own problems, but the top-down approaches are limited in the terms of material systems that we can use. And that is because we cannot deposit thin films of every single material that we know. But also there's other nanomaterials such as liposomes and micelles, so soft materials that we simply cannot produce via these top-down methods. They're also not really scalable, and the cost of fabrication is just through the roof. To give you an example, to produce typical plasmonic structure, you would use about 5 grams of gold, which costs a few hundred pounds, and you will waste about 99% of that gold because you will deposit it in the mask. So it's really not a great method of producing nanoparticles. On the other hand, you have bottom-up bottom-up synthesis where you are able to use precursors that are much cheaper. You are able to use much wider variety of materials, but you struggle a lot with placing these materials in desired locations, or in fact combining them with other systems. So, for example, to have a coupling between two different materials. Nonetheless, there's a lot of interest in using these nanomaterials for many different applications because of the unique properties. So this is the reasoning why a lot of my work was on how do we combine materials together, how do we control these nanoparticles, essentially colloidal nanoparticles, and how do we get them to do what we want them to do. And in this sense, what we want to do is actually to combine different nanoparticles together, for example, or to place them on a specific substrate, both of which involve, firstly, delivery of nanomaterials to a specific location, and secondly, fixation of that nanoparticle, for example, in a specific location. And for this, we have many different tools in our toolbox, not least of which is the long-range attractive forces that exist just about between everything. So if you have two nanoparticles that are far apart, they will experience van der Waals forces, and they will be attracted to each other. But you also have a wide range of intermolecular, intramolecular forces that you can either drive particles together or fixate them to one another. Of course, you can also apply external driving forces such as electrostatic forces, Brownian motion, gravity, optical, convective, capillary forces. So we do have quite many things to work with, but it wasn't until the last 3 decades, perhaps, where we started to understand how to exploit these for actual fixation of materials. I should mention, though, that in the realistic problem involving fixation of materials, you will never be dealing with only one of these forces. Typically, you will have a fairly wide combination of them. For example, in the antigen-antibody coupling, you will probably mostly be dealing with intermolecular forces, but there will be many sites at which these forces are able to act on. But also there's a steric interaction where the two fit exactly together, so the antigen and antibody fit exactly together, and then at every point you can have many of these forces adding up. And so in the end you end up with an extremely strong bond between two materials that, but the underpinning forces are quite weak. Similarly, for example, one of the nanoparticle assembly methods, the driving forces, there's two driving forces, which is capillary and convective forces, and they're strong enough that they're able to overcome the Brownian motion of the nanoparticles. But I will come back to this a little bit later as well. So this is what I wanted to talk about today. Whilst giving some examples from own research where I've used the different methods that I'm talking about. I will start by talking about assembly of nanomaterials in solutions. So how do we combine them with different materials in solutions, or how do we drive them to become something else in solution? I will then talk about the few methods that we have for placing the materials in specific locations and substrates. Before coming back to solutions, but talking now about active control of nanoparticle motion in solutions. Okay, so we'll start with the self-assembly methods, which are essentially the methods we used for controlling the relative placement of two or more nanomaterials when they're still in a colloidal solution. Of course, for these purposes, there's many different techniques that exist. For example, you have the antigen-antibody binding that is extremely widely used in biology, for example, for labeling cells and things like that. You also have something called layer-by-layer assembly, and actually that's now a massive group of techniques, and this is an extremely good review paper that I would suggest for everybody to read about the various ways in which you can fabricate fairly complex structures using layer-by-layer assembly. But in many cases, the assembly of materials is relatively straightforward. So for example, in my research during my PhD, I used the straightforward electrostatic self-assembly to combine biomaterials and colloidal semiconductor quantum dots, with the aim of using these quantum dots as light harvesting elements for the photosynthetic units of the biological systems. So the nice thing about using biomaterials and combining them with colloidal nanoparticles is that they typically have many amino acids or other electron deficient groups, and essentially forming a slight positive charges on their surfaces, which you can then electrostatically attach with the negatively charged colloidal nanoparticles. So that's a straightforward electrostatic assembly with which you can assemble complexes of quantum dots and, for example, in this case, reaction centers. So in this case, we extracted the reaction centers from a light harvesting photosynthetic system of Rhodobacter spheroids. So it's a bacteria that use photosynthesis for energy production. And once you extract these units, you can self-assemble them on the surface of the quantum dots electrostatically. And you can show that the quantum dot is then able to absorb the energy, UV energy, and then down convert it to the energy that's more appropriate for the reaction center. That happens extremely efficiently with FRED efficiencies of up to 80%. So forced resonance energy transfer efficiencies. And that is both due to the very strong interaction between the two, so very, very close proximity of the two elements in this case. We also, in this case, show that the actual emission from the reaction center is in fact improved by the addition of the quantum dot. So essentially, our quantum dots act as artificial antenna when they're for the reaction center when they're assembled together in a complex. I should mention that we only observe the fluorescence from our reaction center because we add antioxidant into the system, because otherwise the reaction center would do its job and to split the exciton into the individual charges. But we constantly provide additional electrons, and so that state is closed, and so we're able to monitor the function of the reaction center by measuring its addition. So that's one of the examples where I was able to work with a fairly complex system by using a very simple assembly method. However, it's not always that simple, as I've discovered when I was working with bacterial rhodopsin protein, which is also a photosynthetic protein. It's the simplest photosynthetic known in general, in that it combines this bacterial rhodopsin protein, which essentially pumps protons out of the bacteria, and that is coupled to the ATP pump, which converts ATP to ADP. Right? So those are the only two components of this photosynthetic system. And the purple membrane, which is a two-dimensional crystalline crystal of this bacterial rhodopsin unit, whose function is to pump protons, and the function is all due to this one molecule called the retinal molecule, which sits right in the middle of the membrane. And this molecule, when it absorbs a photon, undergoes a cyclic sequence of transformations, going through different excited states, which you can monitor spectrally. And that results in a pumping of a proton from one side of the membrane to the other cell. The one thing that made this protein very interesting technologically is that it has very good photoelectric properties and photochromic properties, so you can build a lot of memory devices out of them, or desalination based on their charge transfer properties, for example. It also has been optimized by millions of years of evolution, and so it has incredibly high stabilities in any sense of the word. So you can cycle this system thousands and thousands of times without any loss of function. The one thing it's not able to deal with well is to use UV photons, and that is because it can destroy the retinal molecule itself, so the element that is in the membrane. And because of it, it only utilizes a very, very small proportion of light, and so it's not a very efficient protein in that sense. So from our point of view, then adding a quantum dot, which we already knew can act as an artificial light harvesting antenna, seemed to be an ideal option because they're able to down convert the energy to the energy that is optimum for the bacteriorhodopsin protein. So of course, we went ahead and tried to assemble the system, and this is where we run into a few problems, as you see from this AFM data. So again, this is just electrostatic assembly of quantum dots on top of a bacteriorhodopsin protein. And as you can see, there's no specific attachment of any quantum dots in a general case. Whilst you very clearly can see this trimeric 2D crystalline structure of the bacteriorhodopsin protein itself. However, what we found is that if we had the size of the quantum dots just right, we would actually be able to self-assemble it in a very controllable manner, as I show here. And so what is going on here is that the quantum dots, which are negatively charged, are generally electrostatically repelled from the membrane itself, because it is also negatively charged. However, it is electrostatically attracted to the bacteriorhodopsin units themselves, which do have amino groups on the surface of the membranes. And so if our size is just right, then we are able to place approximately one quantum dot in each of those unit cells, and the electrostatic attraction between the protein and the quantum dot is sufficient to hold them there and to form a very nice assembly of quantum dots. However, in a general case, the steric interactions between quantum dots between each of these units will prevent them from depositing nicely, and so you end up with a more or less disordered organization of quantum dots. Nonetheless, there were cases where we were able to assemble the system very nicely without going to the full chemical conjugation route, which we also did do. But even with the self-assembled systems, you can again see a very nice transfer of energy, and we can see that both in the lifetime quenching and in the fluorescence quenching for the quantum dots. The other nice thing about this protein is we can actually remove this retinal molecule from the membrane. And in this case, the energy transfer route is no longer possible, but all the other interactions between the quantum dot and the membrane are accounted for, and so we can use that as a control for energy transfer measurements. But in general, we did show that quantum dots indeed are able to act as artificial light harvesting complexes, even for this particular system, yielding an improvement in the protein pumping ability of the protein. So in this case, we assembled the same protein into proteoliposomes so that all of the protons would be charged to the inside of these proteoliposomes, and we could just measure the pH of the solution outside to evaluate the pH pumping ability. We also saw a corresponding increase in the nonlinear refractive index of our complexes, and that relates to the photochromic ability of the bacteriorhodopsin protein. Yeah. So I won't go into too much details about this piece of work, but it's just to show that there's other ways in which you can self-assemble materials and using other forces. So for example, in this case, we use the hydrophobic effect, so the hydrophobic interactions of hydrophilic solutions to produce superclusters of metallic nanoparticles. So the protocol for this was very simple. We started with nanoparticles in aqueous solution as they are produced, and we add a dichloroethane, which is an organic phase at the bottom of the centrifuge tube. We centrifuge everything which pulls the nanoparticles to the bottom of the centrifuge tube and concentrates them. We then remove the aqueous phase from the top and emulsify the solution, which creates these pockets of aqueous phase containing many, many nanoparticles. And when we dry them, we end up with these, let me go a little bit further, we end up with these massive clusters of metallic nanoparticles. The reason why we were interested in them is that they essentially become a plasmonic metamaterial where the properties are defined by both the properties of the single nanoparticle, but also by the size of the overall system. So you have a collective modes that are associated with the whole particle and not just individual particles. And the interesting bit about these modes is that some of them do have quite strong fields on the inside of the cluster, as well as its surface. which you can then exploit for sensing. So here we were just confirming the existence of these modes that is essentially internalize the collective modes that we can use for sensing of, for example, protonation or deprotonation of a group on the surface of a nanoparticle with extreme sensitivity using very simple Raman measurements. So I hope this gives you a general understanding of how even self-assembled, very simple self-assembly of materials can lead to investigations of fairly complex systems. And you don't really need to go any further than that when you study interactions between individual elements. However, for a lot of the technological applications, what we want is actually to be able to exploit properties of different materials which are optimum for different applications or for different functions. We also want to avoid any crosstalk between different components. We want to be time efficient, and we want to have a very stable and reproducible system. And unfortunately, nanoparticles, colloidal nanoparticles struggle with that because we, at least 30 years ago, we didn't really have many methods to achieve controllable deposition of these particles on substrates. Luckily, however, there's been a lot of research done in this area, and now we have many techniques to choose from that allow you to localize nanoparticles in interest of location. All of them have advantages and disadvantages, and I show you a few examples here. Like, for example, light-activated molecular immunization is probably one of my favorites, where it uses the plasmonic fields of the nanostructure to break a triple sulfide bond, and that leads to the attachment of BSA on the nanoparticles, which you can then react with another nanoparticle to place it right in the hotspot of an antenna. There's tip-in lithography, photoembolization, cut and paste, MACE-AD. There's many different ones. When choosing a technique that you want, you always have to think about what it is you're trying to achieve and what are the limitations of each technique. So in this sense, to give you an example, in our research, we were very interested in using this two-step EBL method, which is a method for localization of nanomaterials, or quantum dots in this case, in regions of interest near pre-existing structures. As the name suggests, it involves two steps of EBL writing, where the first step involves the fabrication of nanoantenna. And then the second step, you repeat the ABL and you align your write to the first layer. But in this write, instead of depositing metal at this step, you perform chemical reaction to attach your quantum dots to the substrate in the exposed holes. And we do this by formation of self-assembled monolayer on the substrate, and then a reaction between the functional group of the quantum dots with the functional group of the self-assembled monolayer using either antigen-antibody coupling or carbodiamide chemistry. So why did I choose this specific method to work on? Well, because it allowed me to do characterization of waveguides and light matter interactions in a very facile manner. So for example, in this case, we were trying to characterize hybrid gap plasma waveguides on silicon uninsulated geometry. So this is the structure of your waveguide, where you have silicon and insulator waveguide, and then on top you have a plasmonic gap waveguide. And depending on the size of this gap, you can have the mode being fully photonic confined in the silicon waveguide, or it can become fully plasmonic and confined in the gap plasmon. But what the group of Rupert Alton, in this case, wanted to do was to characterize these plasmonic waveguides and actually to prove the existence of the plasmonic gap mode and that it was localized there, so confirm theoretical predictions. And the typical way to characterize these waveguides would be to fabricate many of them of different lengths to be able to obtain the propagation length. So essentially, the coupling efficiency at different cutoff lengths. However, it's much easier to just use selectively deposited quantum dots on a single structure to do the same thing. And in this case, we use the 3-photon emission from the quantum dots with the idea being that we pump in the waveguides at 1500 nanometers. And so our third harmonic of that occurs at 500 nanometers where the quantum dots absorb strongly, and then they emit at 650 nanometers where there's no other signals in our system, so we can very easily measure our fluorescence from the substrate. And indeed, this is what we did. So here you can see these little blobs, which are quantum dots deposited in the waveguide. And this is a CCD image of our waveguide when we coupled the light. So this is the coupling grating. You can see some reflection from it. And then this is our waveguide, and you can just about resolve the 3-photon emission signal coming from the waveguide. So we can then take a cut across this gap and plot it as a function of length. And then the propagation length we then obtain from a very direct fit to the 3-photon emission data. So in this way, we were able to obtain the same information from a single structure as we obtained from tens of structures, and we only sacrificed one of those structures. So if you, for example, manufacturing a large number of these waveguides, you only need to expose them to quantum dots, a single structure, and you will have information about many, many of them. So the second application of the same method was a fabrication of these nanosources where you can fully deterministically control the radiative properties of quantum dots. And the issue with using quantum dots, colloidal quantum dots for this, is that, of course, in a typical sample, you have many, many different sizes present. And so when you try to couple them to plasmonic antennas, you end up with emissions coming from different wavelengths, being quenched, blue shifting, and so forth because of the high electric fields that they're in. And so if you wanted to create sources with very controllable properties, then you're actually better off using something like plasmonic ring cavities, which have much wider hotspots, essentially, where the fields are lower, but you have much wider area where the fields are non-zero. And so in the same method, you can then place quantum dots on the inside of this plasmonic ring cavity to make them couple to the plasmonic structure. And here I'm showing an AFM image of the plasmonic ring cavity, which is 40 nanometers high, which is these two peaks here in cross-section. And there's a ring of quantum dots deposited inside, which is this little blip here in the AFM data as well. You can clearly see that you get emission from the quantum dots, so they're not affected by the process of this deposition method. We then went on to characterize our coupling, and you can see, for example, for an array of quantum dots deposited very close to the rings, you really do get more or less consistent intensity emitted from these structures, as well as a very consistent lifetime. And all of it follows the theory quite nicely as well. But of course, you may say that, well, okay, we're exploiting we're depositing quantum dots, but we're depositing in large areas. But can we actually precisely deterministically place a single particle in a specific location on a substrate? So the holy grail for technology would be making arrays of these nanoparticles. And for this, we also have, of course, many techniques. Each of them have their own advantages, disadvantages. And speaking of which, I cannot, of course, omit the work of a group of Fernando Stefani here in Argentina, who work on optical printing of colloidal metallic nanoparticles. Unfortunately, for this method, though, it only works with metallic nanoparticles because it requires the additional force, scattering force that you get with plasmonic nanoparticles, but not with other nanomaterials which don't scatter radiation as strongly. The second issue with this is that it does require deposition of a polymer or the layer that is charged to provide this electrostatic repulsion from the substrate to provide essentially this energy buyer for the deposition. And the same issue exists with the capillary and conductive assembly of pre-partant substrates, what we affectionately called kappa. And the problem with this additional layer is that you lose the contact with the substrate. So if you wanted to exploit the exact interaction between your nanoparticle and the substrate, such, for example, in photocurrent applications, you may not be able to do that with these specific methods, even if you're using colloidal metallic nanoparticles. That being said, that for a lot of applications, these methods are more than sufficient. But from our point of view, it was interesting to be able to extend these methods to making arrays of nanoparticles, of any nanoparticles on any substrates over fairly large areas. And this is the method that we've developed together with LMU, Imperial College, KAIST, and SUST as well. And this is an adaptation of the capillary-assisted particle assembly method. And in fact, we do use it in one of those steps. But we use a removable, a dissolvable substrate instead. So the way it works is you start with a silicon master template, which then you spin coat PMMA on at fairly low thickness and low concentration, which allows you to very carefully then strip this an inverse of the design with a simple tape. You can then deposit nanoparticles using copper into the holes of these of this template, and then you take this tape and press it against whatever substrate you desire and place the whole thing in acetone, which is then dissolved and hopefully leaving nanoparticles on the surface. Of course, here you get an interplay between the forces you get between the nanoparticle and the substrate and the forces you get between nanoparticle and the solvent. So if the latter are stronger, the particles will lift off, but if you manage to print them strongly enough, then they will remain in the substrate. And using this method, you can really print very, very large arrays of nanoparticles. So these are 100 nanometer nanoparticles spaced, I think, about 2 microns away from each other over a very large area with printing yields of about 98% and accuracy of about 60 nanometers in location. We showed that we can print them on many different substrates, including gold films, ITO films, and gallium phosphide films, and on the PDMS as well. And we also able to show that by comparing the photoelectric current generated from these two samples, that we do indeed get contribution from the hot carriers generated in the plasmonic nanoparticles, which are then able to transfer additional electrons to the ITO. Okay, so far I've covered control of materials and solutions, or relative attachment of materials and solutions, and then their localizations on substrates. And in the last part of the talk, I wanted to go in a slightly different direction, actually to talk about control of nanoparticle motion in solutions. So for that, of course, we do have some techniques already, such as optical trapping, or plasmonic-enabled optical trapping, where we're able to trap nanoparticles in specific locations. And these techniques, optical trapping techniques, generally work in a very, very short range of less than 100 nanometers, or some for plasmonic equivalents, you go to less than 10 nanometer range. So you're really able to control the position of one nanoparticle in a solution with that level of precision. Alternatively, you can go to sort of medium range control using the electrophoretic forces, which are forces that result from the gradients of electric fields produced electrically, and which involve the polarization of nanoparticles. And also, there are very little techniques, however, that allow you controlling the diffusion nanoparticles over very long ranges. Only recently, some of the techniques started to appear on this front, and most of these have to do with the advent of SLM technology, so special light modulators technology, where you're able to control the phase of light at each pixel of the SLM, and so you can start to structure optical beams extremely well, or spatially for the matter. And with these techniques, you can for example, rotate nanoparticles very controllably or make them follow circular geometries. So a lot of this is underpinning the quantum optics technologies measurements. You can also now start to assemble several nanoparticles in solutions very controllably by creating several optical traps using these SLMs, or by switching the pixels in a particular manner. You can, for example, drive nanoparticles to go in a particular place. But as you can see, that all of these techniques involve a very accurate control, temporal control of one piece of equipment. And what we actually wanted is to have a very simple implementation that would allow you to control long-range forces. The reason why we're interested in the controlling long-range forces or long-range transport of nanoparticles is because that typically size-dependent, so it can allow particle sorting. It is can also allow temporary concentration of samples. So if you, for example, perform sensing of a solution of viral particles, if you're able to controllably direct the diffusion of the viral particles to a specific location, you can then sense your viral loads at a very specific location at much higher concentrations. So as a result, you improve your limits of detection. And so on. So this is the reasoning behind going into the control of the diffusion. And of course, diffusion of nanoparticles is nothing else but Brownian motion, which is a stochastic process that results in random motion of nanoparticles as a result of interactions with the solvent. And it's characterized by a mean displacement of 0. So for an ensemble, a particle will have no displacement versus time. However, the square displacement is proportional to the diffusion coefficient, which itself is a function of the size of the nanoparticle and the viscosity of the liquid. So an important thing to note is that Brownian motion occurs in all the systems, at equilibrium or non-equilibrium systems. It's always present. So even if we provide additional forces, such as those provided, for example, in an optical trap, we will still have this thermal noise that is associated with the Brownian motion of nanoparticles. And in optical traps, this is a problem. However, from our point of view, is that we can use this noise for long-range transport as long as we can exploit it somehow. And to exploit it, what we do is actually we throw our system out of equilibrium. Okay. So just to explain how these rectification and Brownian motion works, if you had an array of potentials that are asymmetric in nature, and then you placed a particle in one of them, let's see. Okay, so we trapped a particle in one of the potentials, and then we let us turn the potential off. Our particle would then diffuse a certain distance given by the diffusion coefficient. And let's say that it diffuses in a favorable direction to the next unit cell, and then we apply our potential again, which is asymmetric, so the particle will be driven to the bottom of that potential well. If that is then again turned off, the particle is able to diffuse, and let's say that now it diffuses in the opposite direction, which is unfavorable for us. If it diffuses such a distance that is still within the same unit cell, it will just return to the same minima again. And so in this way, there's only two possibilities really. One is that the particle remains in the same unit cell, and the second one is that it has traveled to the next unit cell, in which case it moves to the next unit cell. And so in this way, you can rectify Brownian motion. One important thing to note is that it's strongly dependent on the time that the potential remains off. And the reason for that is when the potential is off is the only time when the particle is able to diffuse. And if the time is too short, then you will always end up in the same potential minima, so you end up with the trapped particle. Or if the time when potential remains off, then the particle can diffuse in both directions with equal probabilities, in which case you just have Brownian motion type statistics, and you don't have this preferential direction of transport of nanoparticles. Okay. So in terms of potentials that we use, we can really use any type of potential that you can hope to imagine, but being from a photonics background, of course, we would like to use some sort of photonic potentials, and I'm not sure why this is not switching. Okay, there we go. Oh no, go back. Okay, so in our case, we use plasmonic structures for several reasons. One is that it's very, very easy to implement asymmetries in plasmonic systems, and very easy, well, we have a lot of expertise in designing them by now in the group. And so these are the plasmonic, what we call ratchets that we've designed. These are arrays of essentially diamond antennas that is slightly offset from each other in size. And what it does is that at specific wavelength, only one of the set of antennas will be strongly resonant, and the other one will have partial response to the wavelength, creating that asymmetric profile that we're looking for in our potential. And so we can use these ratchets then in a very simple setup involving so our substrate here and then a solution of nanoparticles on top. That could be microfluidic or not, depending on the application. On top, we have the white light source, which we use for imaging, for capturing, tracking the nanoparticles. And this is the only element that we really need to drive our optical ratchets, which is a CW980 laser, which costs 100 pounds, and an optical chopper, so something to modulate our signal at specific frequency. And so the experiments would involve just tracking nanoparticles, many nanoparticles over long periods of time, and then running statistics. So to show you, for example, so this is a control experiment of Brownian motion nanoparticles. You can see that in general, they will have some displacement from 0, but on average, you have absolutely no preferential direction for motion, as opposed to the case where we actually are ratcheting. So we are rectifying Brownian motion, where the direction along the y, velocities along the y direction are a lot higher than the velocities along the x direction. And we've tested it for several different nanoparticles, mostly polystyrene polymeric nanospheres, because they're easier to track. And just to show you a couple of videos, so these are, for example, polystyrene spheres that are undergoing Brownian motion, a little bit jaggedy, but you can sort of see that the particles wiggle around but don't really move. And this is the same when we apply the ratcheting, the ratcheting signal, and you can see them really move quite fast. And these are same spheres but much faster. You can just about resolve these guys. Okay. And then the PTB7, which is a conjugated polymer nanoparticle, unfortunately the ratchets were oriented in the different direction, but generally hopefully I can show you that we're able to ratchet different types of nanoparticles, and really we've tried metallic nanoparticles as well, and they move extremely fast, but they also deposit because they're heavy. And we at the moment trying to track quantum dots, but they're very small and have our camera is not sufficiently good for that, so we're moving our setup to account for that as well. So the one thing to note is that we do expect a frequency dependence for our ratcheting, so it should be strongly dependent on the frequency with which we modulate the signal, and that is indeed what we see. So we do see only this preferential movement of particles for specific frequencies, and we also see dependencies on the laser power. And just to say that there are, of course, other examples of Brownian ratchets. The novelty here is not the rectification of Brownian motion. For example, even there's a photonic example. However, what we do obtain is we exploit the light-matter interaction in a very efficient way that allows us to achieve much higher velocities than those previously reported at much lower power requirements. So at the moment, we're working in implementing the system into a sensing scheme where we'd actually be able to demonstrate the concentration of samples using these ratchets. But as far as this all goes, of course, I would like to conclude, and I hope I showed you that even simple methods can lead to some very interesting investigations and very interesting combinations of the materials. But the last decade has really been an excellent time for those looking to produce more control geometries involving many different types of materials, either to study new physics or in fact to showcase the potential of nanomaterials for technological implementations. Of course, I didn't do any of this work myself, so I would like to thank many different people. In fact, I couldn't fit everybody on this page, so I only thank here the main contributors, and everybody else's names you can find in the individual references. But of course, for main contributors, I'm thanking people who either directly did a lot of the work. So, for example, ratcheting was done by Marciano Palma do Carmo, who some of you may have known, who visited here 4 years ago. Also Alberto Lauri and Michael Nielsen for the two-step ABL work. Emiliano Cortes, who is the main collaborator for the deposition of arrays of nanoparticles. Also some theoreticians, Paloma, Pablo, Francisco Rodriguez, and Alexander Govorov for various different aspects. And then we have Mikhail Artemyev and Nikolai Gaponik, who both grow quantum dots in very, very excellent quality. And of course, Stefan Meyer for guiding me through the early stages of my career and really being an excellent mentor. I have to thank the funding and especially the Royal Society, who are the reasons I'm here, and I get to collaborate and come here every few years, or at least did for a while. And I thank you for your attention.
Bueno, ¿alguna pregunta?
Hi, thanks for sharing these nice results. About the ratcheting, so you showed you can move things in one direction. Can you also, do you think you can also turn them? How close you can actually do this channel? Is there a limit to how close you can go? And also, I guess at the end, the idea is not to just move things, but also to order them. So you finally have to be sure that you separate them or they get in a row and they don't overlap with each other.
Yeah, okay. So there's several questions there. In response to your first question, so the dimension over which we can transport them is only limited by the size of your beam, essentially, and the size of your array, right? In terms of technological implementation, that's easily solved by building in the LED system right into the substrate, so you can have an organic LED that illuminates your ratchet, which is right on top. In terms of the ratchet itself, we're working on making it using the self-assembly methods as well by either kappa method or something else. So in principle, that could be extended to very long distances, but you say that's just a one direction type transport. For transport along different directions, it's easy to build a ratchet that concentrates the particles like in opposite directions, if that makes sense. And that's for concentration purposes. But if you wanted to have them change directions, that's where the problems start to appear, because if you're using a polarized light source, then you will have the ratchet has to be oriented specifically relative to that polarization. And if you're using non-polarized light, then you can have contribution from different modes of your system, and then so your ratcheting will not be as efficient. But in principle, you can still do it. So actually, we're thinking of using circular polarized light for that, so that you always have an optimum, at least at some angle of the polarization, you'll have an optimum excitation of the system. And in response to your particle sorting question, we actually do have that already built in, because different particles will experience a different potential from the same ratchets. So by driving, and that determines the frequency with which you need to drive the system. So if you, for example, for small particles, you need to use very low frequencies, and for large particles, you need to use much higher frequencies, order of about, I think, factor of about 15 difference. So you already can drive just the small particles or just the big particles using the ratchets that we have. So you already have a particle sorting built in through the frequency selection. Does that answer the question? Yeah. Okay.
Hi, thank you for the lecture. I want to ask you, did you consider another geometry for the case of the ratcheting again? Particles with different morphology, let's say cylinders, for example, or other configurations. And second, in this case, do you expect for them to do other motions, for example, rotating in place or something like that? Sorry for the question, yeah. It's a little bit convoluted.
I mean, okay, in response to the second question, you can build a ratchet that closes in on itself and circularly polarize it, and then that would just move particles. But there's much easier way to do that using electrophoresis, so or in fact SLM technology. So I don't think the system is the best way to go about it. And then in response to your first question was different shapes of the non-antennas. We did consider that, we considered triangular shapes. So we need an anisotropy in geometry essentially, and we did consider different types of triangular systems where we would achieve the same thing. But what we find is that we'll lose that strong coupling between fairly narrow structures. So as soon as you extend the plasmonic structure in the other direction, you actually, the quality of your surface plasma decreases, the quality of your resonance decreases. So in the end, these dimeric antennas presented the best case. If I were to go further with this and optimize the system further, I would actually use machine learning because I think it's the optimum system for that.
Oh, thank you.
Any other question?
Yes, so in the SOI waveguide, I couldn't follow the argument on why a single quantum dot saves you from the trouble of making different geometries.
Okay.
So just if you could explain me about this experiment again.
Okay. We had more than a single quantum dot. We have many quantum dots deposited, but over a single waveguide. Right? So here we expose I think the whole waveguide actually, and so the quantum dots are deposited everywhere, including in many quantum dots that are in the gap of the gap plasma, right? And then we couple our 1500 nanometer radiation to the waveguide, at which point the quantum dots, if the mode is confined in the gap and not in the silicon, the quantum dots will be excited through the free photon absorption process. And then they fluoresce at the natural 650 nanometers, which we're able to image with our CCD camera. Right? So here, for example, there's a cross-section showing this little blip is the one from our coupling grating. And then after that, we have 3-photon emission absorption, 3-photon emission from quantum dots, which is the fluorescence we get from the quantums in the gap. And then we just fit that to get the propagation length.
Okay, so it's fully dependent on how the quantum dots deposit on the channel. It's just, you have to find the right ones to get you different couplings, essentially.
Well, there's many of them in the channel. So we made sure the concentration was high enough that you get a fairly uniform amount of them throughout the whole waveguide. Yeah. And I mean, we always image, the quantum dots are tiny, and we always image in diffraction limited, so yeah.
Any other question? I have a question. When you were using the quantum dots as antennas for rectification, so the idea was to shine them with a particular kind of light and then get the emission of another region of light?
So there were two different systems, two different reasons. So for the reaction centers, we really just wanted to demonstrate the concept that quantum dots can act as artificial light harvesting elements, in that they do absorb light very efficiently over the visible range and the UV range, which is where the solar radiation is. And they're known to be very efficient at energy transfer, so why not try them in the actual photosynthetic system? So that was for reaction centers, but for bacteriorhodopsin, its structure is such that it cannot absorb UV photons, and if you it evolved that way so that it couldn't have a light harvesting system that absorbs at those energies. So that was our idea, is that try to use that as an artificial light harvesting element that in fact absorbs UV radiation, then through the excitation processes, the quantum dot relaxes to the lowest excited state, which is emits in the visible. So then the energy that the bacteriorhodopsin receives is then already down converted to an energy, to a wavelength that is optimum for it. So you get no damage to the protein in the sense, even if you excited UV, but you gain efficiency in that spectral region. But of course, the interest is that bacteriorhodopsin is actually a very technologically relevant material, so if you're able to improve its efficiency and its response to solar radiation, then using quantum dots, then that could be a way forward to implement it in technologies.
And the last question, in order, when you were trapping your quantum dots inside the rings, that was an effect of a plasmonic-driven interaction, or it was a two-step EBL process that you would cover the outside of the ring and then put them inside?
That was a two-step EBL.
Okay.
So in this case, we could controllably control the distance between the quantum dots and the plasmonic ring cavity by exposing different rings, patterns essentially, that are aligned to our plasmonic ring structure. So the plasmonic ring is quite large, it's about 400 nanometers, so we had some room to play with.
Thank you very much. Well, we thank Dr. Rakovich. Y tenemos unos anuncios.
Gracias. Bueno, no, quería comentarles ahora, entonces tenemos intervalo para el almuerzo entre las 12 y las 14. Para almorzar les pedimos que tienen que salir por la misma guardia que entraron y en la zona del gimnasio donde nos registramos va a estar la posibilidad de almorzar adentro del gimnasio o en los food trucks que están por ahí cerca. Va a haber 2 food trucks ahí también. Eso respecto del almuerzo. Siguiendo con los anuncios gastronómicos, quedan a la venta como unos 100 lugares para la cena del jueves, que los estamos vendiendo ahora y durante la tarde en la secretaría de la reunión, que es aquí nomás saliendo a la derecha. Y entonces también les quería comentar sobre las actividades de la tarde. Empezamos a las 14, entonces vamos a tener las reuniones de división que van a ser la mayoría acá en esta zona del pabellón Guido Beck o en la ex biblioteca o en aulas nuevas. Y vamos a tener en la región sur del centro atómico 2 divisiones, la división de partículas y campos y la división de industria y tecnología. Para eso, quienes quieran caminar, son 10 minutos de caminata dentro del centro atómico, el clima está lindo, pero también va a haber algún micro de la Comisión de Energía Atómica que los puede llevar a partir de las 13:30, 13:40. Y el último anuncio, creo que también a las 14 vamos a abrir el lugar para poner los pósters de hoy, los pósters del martes. Ese es el edificio de telecomunicaciones que también está en aquella zona, o sea, más o menos 10 cuadras de aquí. También los que quieran ir caminando, se puede ir caminando, o si no, alrededor un poco antes de las 14 vamos a tener el micro que los lleve. ¿Está bien? Eso termina a las 18:30 aproximadamente, va a estar abierto hasta las 19. Y para los que hayan venido en micro, los micros van a salir de aquella zona, de la zona sur, para que no tengas que volver a caminar hasta aquí. El último anuncio es que, bueno, mañana casi todos ustedes están registrados, pero si hay alguno que no tiene credencial, mañana vamos a seguir registrando a la mañana en el gimnasio y a partir de mañana también vamos a estar pagando las ayudas económicas. Así que bueno, creo que eso es todo. Muchas gracias, nos vemos a la tarde.